Hi, this is Neeladri from Professor Christine Orango's group at UCL. In this poster, I will be talking about characterizing and explaining disease-associated human mutations in proteins without available protein structures or homologues. This work was carried out in collaboration with David Baker from University of Washington and Samir Velankar at the EBI. Over the past few years, there have been massive improvements in deep learning-based protein structure prediction. The models of the protein structure can help explain disease-associated mutations based on proximity to functional sites, destabilization, and pathogenicity. We have created a database of such disease-associated proteins from var site, and we used HamsaVar for the disease-associated mutations. For our study, we used models built using two state-of-art deep learning-based predictors, AlphaFold and RosettaFold. We chopped these proteins up into cath domains. Then the remaining regions were assigned to PFAM, whereas the remaining regions which couldn't be assigned to either cath or PFAM were being called unassigned domains. We noticed that the AlphaFold models were of better quality compared to RosettaFold. However, we had 227 and 45 cath, PFAM and unassigned domains respectively, where RosettaFold was of a better quality. We structurally superimposed AlphaFold 2 and RosettaFold models and found that cath models had lower RMSD compared to PFAM and unassigned domains, which had the highest RMSD. As expected, with an increase in RMSD, the quality of the models also reduced. We noticed that more cath domains were of better quality compared to PFAM or unassigned domains. To explain this, we checked the multiple sequence alignment that would be used to build the models and also the percentage disorder in these domains. We saw that the cath domains had low percentage disorder and had higher diversity of the multiple sequence alignment that were used to build the models. The diversity was calculated using the NEV values, which is the number of effective sequences in the alignment at 80% sequence identity. The unassigned domains had the highest percentage disorder and lower diversity in their alignments. We explained disease-associated mutations in these proteins based on proximity to functional sites, which were predicted as ligand binding site, protein-protein interface, or conserved sites using P2Rank, MetaPP-ISP, and ScoreCons. We also checked if these mutations were structurally destabilizing or pathogenic, which was predicted using FoldX, Dynamute2, and MutPred2. We saw that we could explain about 80% of the disease-associated mutations in good quality regions of the proteins, as seen in the pie chart, based on either proximity to your predicted functional sites or destabilization or pathogenicity. We had higher confidence in the predictions when multiple tools predicted the same site as on or near a functional site, which can be seen in the Venn diagram. Usage of RosettaFold models, we could additionally explain 93 mutations which could not be explained using RosettaFold AlphaFold models, because the AlphaFold models were either of low quality or the predictions could not come from the AlphaFold model. Around 62% of the mutations predicted on or near functional sites in AlphaFold models were also predicted to be on or near a functional site based on RosettaFold models. We have higher confidence in these predictions. We have a few structures where we could explain the disease-associated mutations in red based on the proximity to ligand binding site or a protein-protein interface, as can be seen by the gray colored surface representation of the three proteins in here. We also compared the disease-associated mutations to polymorphisms. We saw that disease-associated mutations are more buried, structurally destabilizing, pathogenic, and closer to the predicted functional sites. With this, I come to an end. We have higher confidence in the predictions if both AlphaFold and RosettaFold predicts a site to be closer to a functional site. We also have higher confidence in those predictions where more than one predictor is telling it to be a functional site. We also have higher confidence in those predictions that are coming from models where both AlphaFold and RosettaFold have low RMSD. That means two different tools are saying that that particular protein sequence is taking up the same are taking up the same structure. Hence, we have higher confidence in those models. We have already published this work in Briefings in Bioinformatics, which you can scan the QR code to get further details. Thanks a lot for listening to my talk.